trying to maybe have a different answer and stuff like that. Sometimes when we are discussing, people have some different questions and answers, or yes, let me know so that at least we go together. The reason why I'm doing this is because this is not the last time we're gonna be writing life science. So we have still the prelim, we still have the final exams. So we need to know our mistakes. We have done those slight mistakes which are going to make us lose the marks, and then we are able to at least focus on high marks. All right, let's start without wasting time. It was June examination, grade 12, 2024. Life science. Don't forget that this paper had both paper 1 component and the paper 2 component. The first question was clear. Guys, as I told you that sometimes you have challenges when sometimes when I'm also doing, sometimes even I skip a question when I'm not seeing it. So please let me know in the comments so that I don't skip, because this class or this discussion is live. All right, let's start without wasting time. They're saying that question, okay, not going to, it's based on basically question. This question to this question is basically based on the diagram below. If you look at that diagram, is a reflex arc. So they're saying that the correct order in which impulse travel in the diagram above is, it starts from the receptor to the sensory neuron, the interneuron or relay neuron or connector neuron to the motor neuron. So we have to look for that. We call it the path. So basically the answer becomes D. Why? Because you can't start with the receptor to go to the interneuron. So that's what makes it wrong. The receptor, then the effector. You can't start from the effector and then to receptor, then effector. Then goes out, then the receptor, then motor neuron. Still, they are not connected in any way. Therefore, the most correct answer is D. So you get those 2 marks. Then they're saying that the path shown in the diagram is known as a reflex arc. Is a reflex arc. Remember, we said that is a path traveled by the impulse during the reflex action. If you remember our last minute discussion. So the answer becomes C. So that one becomes the most correct answer. Guys, don't be sad if you find out that the answer you wrote is different. Why I'm saying that is, don't be discouraged, because you still have other chances, the big chances for you to change your mark so that you are able to focus and get a distinction at the end of the year. You understand? The journey is just beginning. Yes. So don't, at least once you get it wrong, at least you won't forget it again, maybe if they bring it again in another paper. Which part of the brain is responsible for interpreting hearing? This is a sensation. Whenever I talk about sensation, we're talking about the biggest part of the brain, which is the cerebrum. So the answer becomes, the most correct answer becomes B. Yes. So cerebellum is basically equilibrium and the muscle tone. Then corpus callosum is connection between two hemispheres, allows communication. Medulla oblongata is the involuntary actions. That's basically, it's next to the spinal cord. So I'm not going to discuss that much more, because we are trying to see at least the memorandum, and I'm not about giving you the correct answer. I just have to highlight a small bit of it so that you are able to understand if they bring it again. Yes. So you give me that slight time which might deviate from our normal time. So they're saying that in the guinea pigs, did you know that these are not pigs? These are rats. All right. Guinea pig, one parent had black fur, and another one had white fur. Then the resulting offspring we had gray. The moment you have black, and then white, and then you produce gray, then automatically it is an intermediate. Therefore, we say it is incomplete dominancy. You understand? If I had a mixture of black and white, then it would have been codominancy. So in this case, it is incomplete dominancy. So the answer, they're saying that this interaction between F1 allele resulted in gray can be referred to as incomplete dominancy. So B becomes the most correct answer. Then we go to the next question. They're saying that which one of the following is an example of a chromosomal mutation? When we talk about chromosomal mutation, based on our level, we have two types of mutation. We have what we call the chromosomal mutation. We have what we call gene mutation. Gene mutation is as a result of just a base being maybe replaced or substituted or deleted or what. But in most cases, we see it in DNA. You understand? When it's transcription translation, that is gene mutation. But when the whole chromosome change, for example, you have an extra chromosome like Down syndrome, yes, Klinefelter syndrome, Turner syndrome, that is as a result of an extra chromosome. Therefore, because it is an extra chromosome, therefore we call it chromosomal mutation or chromosomal aberration. So in this case, they're saying that which one of the following is an example of chromosomal mutation? Albinism is a gene mutation, colorblindness is a gene mutation, hemophilia is a gene mutation, just a base has been replaced. So the only answer there is what you call the Down syndrome. I think those people who have been following us, I think you got these answers right. If you didn't get it right, I know that you just forgot them. Ne? Yes. So just keep on following us on our YouTube channel, TikTok. You'll be able to, and Facebook, you'll be able to at least to benefit in your discussions with the papers or making your life a little bit simpler. All right, let's go to the next question. They're saying that a farmer is growing pea plant. Pea plants, you know them, you buy them from ShopRite, ne? ShopRite, Pick and Pay, whatever. Ne? The allele for tall is capital T, and the allele for dwarf is small t. The moment you say capital letter and small letter, definitely that is one is completely dominant over the other. Which cross would need to be carried out to ensure the phenotypic ratio is 1:1, meaning that I have to get the black and I have to get the white. So in this case, it cannot be, you cannot cross capital T completely, never, with the capital T. You can't. You can't cross capital T and small t, because all of them, they're going to be one color. So definitely it means that one must be at least heterozygous. So what if I cross capital T? Because here, if I cross, check, it's going to be, look, you see that all of them are going to be the same thing. You see, going to be, when they bring such questions, try. Don't just give the answer. Ne? Don't just give the answer, guys. Ne? Yes. All right. So let's cross. So this one is out. If I cross this, I'm going to get a ratio of 3:1. So check. It's going to be capital T. It's going to be capital T, small t. It's going to be capital T, small t. And then it's going to be small t. So this one is also out. Even this one is going to be the same because I have capital letter here. It's out. What about this? Let's test it before we conclude. Capital T, small t. So I'm going to be this with this, capital T, small t. This with this, capital T, small t. Or this with this, small t, and then this is this, small t. So I'm going to get a ratio of 2 to 2. You understand? So if I get a ratio of 2 to 2, what does it mean? I have to make it in the simplest way, because it's the ratio. It's supposed to be in the simplest way. Divide by 2, I'm going to get 1:1. So the correct answer is, that's what they are looking for. The correct answer becomes D. So it means that that is the most correct answer. In this case, our correct answer is going to be D. And I think you see how, guys, when you get such questions, make sure that you practice, you do all of them. You try them. Don't just give the answer. Try all of them so that you are able to understand whether everything is correct. All right, let's go to the next question. If you have any question, complain, let me know now, because this class is live. Ne? Yes. Then I say the microscopic structure that carry out the microscopic structure 1.7. The microscopic structure that carry out part of protein synthesis under the control of the nucleus. They're saying microscopic structure, and they're saying that carry out part. This is the key point. It's a part just. Ne? So it can't be cell wall, it can't be mitochondria, because under the nucleus control. So these ones, all of them, because remember they have DNA, so they're out because we have the nucleus there. So the answer is the ribosome. As you getting it right, né? Blood group A, BO, the ABO blood groups are inherited characteristic. What would be the possible blood group of the offspring? B, when the parents have genotype of this. You have this. Okay, now you have also to practice and see. Don't just give the answer. You understand? So this with that, you're gonna be A, that with this, sorry, that with that. This one is out. It was just a mistake. That with that, so it's gonna be A. Then this with this is gonna be A, homozygous, and then this with that is gonna be still A, heterozygous. Two heterozygous A, two homozygous A, and two heterozygous A. So all of them is gonna be blood group A. So I have to look for what is A only. So the answer becomes D. So D becomes the most correct answer. We go to the next question. 1.1.9. What are the receptor in the ear responsible for maintaining balance called? How do you call that? It's the cristae and the maculae. If you follow the Saidi's model of balance, I think you get it right. Yes. So it's the cristae and the maculae. So that is the most correct answer. Then they're saying that 10. The diagram below represents what is it? What do you see here? You see it's a diploid, then you have haploid, but these cells degenerated. You understand? So you see that this one degenerated, also this one is degenerating. So how do we call this? What is this? So it's under the influence of meiosis, but what are you trying to do? Check here. Check here. You have 2N, 2N. You see? You form 1N, 1N. You see? So you have 1N, 1N. But they're trying to show you that this one, a small sperm, is fertilizing it. You understand? A small sperm is fertilizing it. What is this? So in actual sense, it forms what we call the ovum. Form what we call the ovum. So the process by which ovum are being formed is called oogenesis. So the most correct answer in that regard is the oogenesis. Guys, am I clear? Do you understand? Are you getting it right? Have you got 20 out of 20? Let me know you have got 20 out of 20. Yes, let me know, guys, if you have got 20 out of 20. Tell me, how much have you got? If you got more than 10, let me know in the comment section. Yes. Thank you very much. Let's go to terminologies. I like them. I like the terminologies because they teach us science. There is no guessing. You either know or you don't know. Attachment of the developing embryo in the uterine wall or lining. We call it implantation. Implantation. Yes. They're saying the manipulation of biological processes. When you say biological, so you tell us bio. It's biological. And the manipulation, yes, of biological process to satisfy human needs. So we call it biotech. Technology. By technology. Yes. The type of inheritance where both alleles are equally dominant. So if they're both equally dominant, yes. So how do you call that type of dominant? They're not that one brings 50 and the other one brings 50. Then you form an intermediate. But here both of them are equally dominant. Blood group A, allele for A and allele for B, when you combine them, you form blood group AB. So they're equally dominant. In this case, we call it co-dominant. Co-dominant. So the type of inheritance, so we call it co-dominancy. Co-dominance. Yes. They're saying that the point of crossing over between two homologous chromosomes, we call it chiasma. chiasma. They are saying a point, not points. So I can't say chiasmara. You understand? Then they're saying that the diagram show below indicates the diagram showing the inheritance of the genetic disorder over many generations. So we call it pedigree. Is a pedigree. The process whereby I is adjusted to the amount of light. How do you call it? We call it pupillary mechanism. Pupillary mechanism. They are saying that the part of the ear that equalizes pressure, the part of the ear that equalizes pressure on the either side of the tympanic membrane. Yeah, they're saying the bundle of nerves that connect the brains to hemisphere. We call it corpus. I always tell people, and some people they say corpus luteum. Unless you you have your how do I call them? Yeah? Your reproductive part is in the brain. Ah, yeah, yeah, okay. It's corpus callosum, corpus callosum. Yes. So guys, basically I think you got 8 out of 8 when it came to that point. Yeah, how many people got 8 out of 8? Yeah, guys, let me know. Don't be shy. Don't be shy. Okay, who got 6 out of 8? Yeah, don't be shy. Talk to me. Talk to me so that I become also happy that oh my kids are getting it. All right. Anyway, I hope you are getting it right. They're saying the structure that give rise to the spindle fiber during meiosis and mitosis in animal cell. The structure in animal cell is the spindles. Sorry, the centrioles. However, né, because they say the structure that give rise to spindle fibers, because we don't have spindle fibers. Did I say animal? Okay. Give during meiosis and mitosis. Oh, in the yeah, it's fine. I didn't talk about the plant cell. Thought that they talked about a plant cell. So it's animal cell, not plant cell, because plant cells they form what you call the plates. Yeah. So they are in this case is spindle is centriole. So the answer is not A. The answer is A only. We want the word only. Yes. Then they're saying that the possible gamete in the dihybrid cross. In this case, is the possible gamete in the dihybrid cross. When you talk about this, we are talking about, guys, please do not do, because it's a dihybrid. So you need to have more than many what? Many uh, gametes. So in this case, it looks like a gamete. I think here there must be a discussion about it. There must be a discussion about it. Must be about it. They are saying that uh this is, if you look at this, guys, you'll find out that this and this is not separated. You understand? This is a genotype. So it would have been capital H, small b, maybe capital H, capital B, yes, small h, capital B, small h, small b. So indeed, that's what you're supposed to if those are the gametes we are talking about. Those are the gametes for the dihybrid. So in this case, this one is for monohybrid. So in this case, the answer becomes none. There is nothing here. Yes. So that's how this is a genotype. They didn't say the genotype, they say the gametes. So these are the gametes for the dihybrid from this genotype. So that's it. An allele of blood group. Yes, alleles of blood group. They're saying complete dominancy and then co-dominancy. Guys, here I would say it's both. But in this case, they are not talking about sorry, I would say is both if they're talking about the genotypes. But in this case, guys, the question is asking alleles. So you have allele for A, allele for B, and allele for O. You understand? So if I have A and B, you understand? So what happens? Then it is codominant. But if I have allele A and that for O, check what is here. This is completely dominant. So it is A. If I have B, like that, what is that? It is completely dominant because this one is completely dominant over this one. You see, so it's complete dominant. So a lot of blood groups. So in this case, there is a discussion about it. They must do the discussion about it. Yes, they'll do the discussion about it. But in this case, I'll say that is both A and B. Both A and B. Because if why codominant in this case, if it is A and B, definitely that is codominant. But if you are talking about, but don't forget that these are genotypes, but they are talking alleles. But alleles are the one which form the genotypes. So in this case, if you look at this and this, you form completely dominant. You see, so it's complete dominance, and then this one is gonna become if it's like this, it's gonna become codominant. You see, so in this case, and what about when it's like this? So here the dominancy we can't talk about it because there is nothing which is different. This is just a homozygous recessive. It's homozygous recessive. So the answer here becomes both A and B, but is subjected to be on discussion. So the location of DNA is in this case is both A and B. They're saying location of DNA. We have the location of DNA in mitochondria. We have the location of DNA in the chloroplast. Yes. Yeah, guys, are you together? Have you got all the 8 marks in this regard? Don't forget that it's both A and B, and then we give you 2 marks there. This word none only, guys, make sure that you do those things when you go for the paper. Yes, diagram below. Let's go to the next question. Yes. 1.1.1.4. The diagram below shows the embryonic development in the birds. Then the type of embryonic development shown in the diagram. In this case, it is an amniotic egg. Ne? Amniotic egg. They brought even this question in the final exam for May, June, but they didn't bring the egg. They just brought. Guys, make sure that you practice different questions before. If other people have written before you, do different questions and then try. And then ThunderEDUC channel is always giving you last minute predictions and stuff. Everything is always there in all sets of exams. So we are here to serve you. You understand? So that you guys, you change your mark, you get high marks, and it is possible. Eh? Give the letter only. Here they're saying letter only. They don't want your name. Yes. But for the case, for the purpose of revision, we will give the name also. Yes, that now shows the embryo. What now shows the embryo? It is the yolk. It's the yolk. But remember, based on our examination guideline, we say yolk sac. You understand? So it's a yolk, yes, because the yolk which has the stores the nutrients, but in this case we say yolk sac. So in this case, what is the yolk sac in there? Based on my diagram, I see a yolk sac as A. So maybe we shall say that that is a yolk sac. And then stores and removes wastes. What stores and removes wastes is allantois. Allantois. So I will say allo toys. So in this case, based on what I see here, alone toys is, it looks to be this. Yes, this sac. So it becomes B. And then it protects the developing embryo. So this one is the amnion. Yes, actually, it protects, it's the amnion. So it's amnion, but in this case, if you look at it, is an amniotic sac, but we call it amnion. You understand? So in this case, I will say it is, if you look at it, I will say that it is C. Yes. So amnion is the one which is responsible for that. So you guys, yeah, that's it. State 2 physical characteristics of precocial chick upon hatching. Precocial, born prepared. Precocial, prepared. You understand? So meaning that in this case, you're gonna have to be prepared. Eyes are open. Eyes open. The eyes are open, meaning that they can see properly. Number two, you can list all of them. They have down feathers, covered with down feathers or in down feathers. Down feathers. You understand? Then you can talk about they can walk, they have ability to walk. They can walk, yes, and they feed themselves. Yes, you see. So those are some of the, they want to. Any other you have written, as long as correct, you get a mark. Yes. All right. Let's go to this question. Many people were complaining about this. Generally, how was the paper? Many people were talking about this is the endocrine system. But if you look at critically, it's not endocrine system. However, remember this part is linked to endocrine system. So it's not endocrine system, guys. A learner code, it is sympathetic and parasympathetic nervous system. If you check on our distinction material book, you'll find out the answers are there. Rana is called while banking from his life science class. A teacher yells out to him to go back to class. He knows he is going to be in trouble, both at school and home. Eh, he considers going back to class, but as the teacher approaches, he decides to run away. The diagram below shows an organ in the body representing or responding to the sympathetic nervous system. So this person took off so that he's safe. I don't know. Describe how the learner's sympathetic nervous system helps to escape with regard to the following organs. So it's just like that. So in this case, the pupil, we talk about the pupil. So when we talk about the pupil, it dilates. You understand? It dilates. The heartbeat, we talk about the heartbeat. Heartbeat increases. Yes. Liver. When you talk about the liver, it converts glycogen to glucose so that you have more energy in the muscles. Yeah. Explain the role of the gland attached to the kidney to play in the sympathetic nervous system. In this case, this is the gland which produces adrenaline. Adrenaline. So it produces adrenaline. What's the function of adrenaline? Basically, it helps in triggering or working together with the sympathetic nervous system. You understand the sympathetic nervous system. So now you bring the individual to to emergence. Yeah, so it works together with sympathetic nervous system, or it prepares the sympathetic nervous system for emergence, or the body for emergence. Yes. So then name the process that will increase in the cell during sympathetic nervous system. I will not comment about this question, but I think you guys, based on grade 11 work. Now I'm seeing that they are starting to bring some grade 11 work too. So I will say, if I'm too scientific, I will say glycogen But if I'm not that too much deep, ne, I will say metabolism. Metabolism. If I'm too much specific, it is glycolysis. But if I don't want, because metabolism doesn't mean only glycolysis, doesn't mean only respiration. You understand? Yes. So it can be aerobic respiration or anaerobic respiration, depending on how much oxygen is present. So basically, that's why you talk about glycolysis. And the function of this is to produce ATP. So when you talk about metabolism, you are now generalizing, including that. So it's better to say maybe metabolism. Yeah. But the actual process is glycolysis. You understand? So I hope they will give the metabolism and they will give the glycolysis. Identify the purpose of the increase in the process mentioned above. Is here is just to produce more ATP, to produce more ATP. So in this case, I will say that this word more is very, very, very important. So it's produce more ATP. Name the branch of the nervous system that allow the learner to calm down once he is far from the teacher. That is the parasympathetic nervous system. I'll say PNS. Parasympathetic PSNS. Parasympathetic nervous system. But you're supposed to say parasympathetic nervous system. You can't just say PSNS. Thank you very much, guys. I hope you got 50 out of 50. How much? There are some questions which were a little bit tricky. Yeah. Guys, how did you see this? Compare Gauteng, Northwest, Limpopo. Yes, they usually say that Gauteng papers are hard. I don't know why. Let me ask you guys. Examiners from Gauteng, why are you making papers too hard for the kids? Because most students say that other provinces, they have easier papers. Their papers are a little bit easier compared to Gauteng. Why? Maybe they're trying to prepare you incoming toughness. Ne? Maybe. So guys, just comment your there that how was the paper? Tell us whether the paper was easy or it was very difficult for you guys who have written this paper. Thank you very much. Let's go to question number 2. Look for another 50 marks. The diagram below shows the developing fetus in the uterus. Name the hormone that is produced by the structure A. Structure A. Structure A. What is structure A? Remember, in our last minute discussion, we talked about that structure A. Structure A is a placenta, and this placenta, when it is above 12 weeks, why is it not writing now? When it's above 12 weeks, okay, when it's above, that is a placenta. When it's above 12 weeks, as lurium will stop producing progesterone, and then now this structure starts to produce progesterone. So now, guys, name the hormone that is produced by structure A. Definitely, we shall say that that hormone is progesterone. Is it clear? Yes. Describe the consequence for the fetus if structure A produce insufficient amount of progesterone. How many marks? Is 2 marks. They're asking you what will happen. Then it means that pregnancy will not be maintained. You understand? And then this might lead to a miscarriage. Maybe we can say premature birth. It can lead to a premature birth. You understand? Yes. So pregnancy will not pregnancy will not be maintained. You understand? And this will lead to a miscarriage. A miscarriage. So we're gonna have what you call the miscarriage. Yes, miscarriage is gonna happen. So because the function of progesterone is to maintain pregnancy, and then now you have reduced the amount of progesterone, definitely miscarriage is most likely to happen. The fetus lung are not fully developed. Yeah, we know, not fully developed for gaseous exchange during the gestation period, during the period of the development of the embryo. That's the meaning. Give the name and the letter of the part that serves as path for the transporting the gases from the mother to the fetus and vice versa. So they sing the letter and the name and the letter. So in this case, we have to look for the letter and the name, which, so the answer becomes B. B, that's the letter, and then in the name is umbilical cord. Guys, umbilical cord is not A. It's not A. Many of people write A. Ngek. It's not A. Is U. U. Umbilical. Umbilical cord. Guys, if you forget the spelling, call them the way they are. No one will know that you called it like that. If you can't remember this umbilical is U, call it umbilical, so that you remember. You have to be with the technique for you to remember these spellings. Yes. All right. So we are done with that. So the answer is B, and then we say it is umbilical cord. Explain how the developing fetus would be affected. How is the developing fetus would be affected if the membranes of structure were teared? Structure C. We come back here. We look for structure C, which is the amnion. What will happen if the amnion is broken? Number one, let me not write so that I'm rushing to finish up. Yes. Number one is infection. Infection, it can lead, okay, you can make this one last, but definitely there will be infection between the infections between the baby and the mother. They brought this question some time back. Yes, it was a final exam. Number two, premature rupture of the membrane. Actually, we call it the premature rupture of the membrane, the prom. In the medical language, we call it prom. Premature rupture of membrane leads to premature birth. I'll call it prom. Prom. Premature rupture of membrane can lead to the premature birth. You understand? Yes, you're gonna produce a premature baby depending on what is the age. You understand? Yes. Number three. So it depends on what you wrote. Yes. Because amnion is responsible for protection, and then now you have messed it up. What happens is there will be reduced or decreased protection. Decreased protection. You understand? Yes. Then you can also, there are so many things you can talk about. Maybe you can talk about maybe compression of umbilical cord affecting the blood and nutrient flow. If the thing is what is now the fluid is not there, this umbilical cord is gonna be compressed, and then that will affect the blood flow and the nutrient flow to the baby and to the mother. You see, so basically, and others you have written depending on our, if they make sense, yes, because here I'm just giving you the most likely answers you could, and then you feel so that you are feeling comfortable. We are the open ones which we are going to allow during the marking session. Menstrual cycle. I know you don't like this, but I told you, if you don't like it, how will it like you? If you don't like me, you think I will like you. I won't like you because you don't like me. So it doesn't like you because you don't like it. At the end, it's easy to pass. Let's see. The diagram below shows the hormone control of the menstrual cycle. Hormone control of the menstrual cycle. Guys, are you in class? Are you here? I don't see any comment. I don't see people talking, or I don't see anybody. Ne? Then why did I make it live? You people, you must talk, you must talk. Yeah, I see now a heart flying here. Thank you. At least now, guys, don't forget to subscribe and like. Ne? Yes. All right. 1 to 7. Name the pituitary gland hormone which showed a sharp increase in the just before day 14. That is LH, luteinizing hormone. That's luteinizing hormone. Someone is yeah. Guys, don't forget, guys, to like, ne? Like, subscribe. Don't forget to do those things. Those things are the one which makes us stronger. So, people like the thing, ne? Once you like, guys, we're gonna make more content. Yes, yeah. All right. Hey, day 14, we said it's luteinizing hormone. Make sure that the spelling is correct. But if you wrote LH, we'll give you a mark. Why? Because this is not a terminology. Describe the function of the hormone mentioned above. This luteinizing hormone, it has two functions. The first function is, what is the first function? The first function is triggers ovulation. Number 2, it changes the empty Graafian follicle. This one, the one after is empty, to corpus luteum. Empty Graafian follicle to corpus luteum. Simple. Number 1, triggers ovulation. It doesn't cause ovulation, it triggers ovulation. Stimulation, it stimulates the Graafian follicle to burst open, basically, so that the ovum is released. That's what we call the ovulation. Yes. Number 2, it stimulates the conversion of the empty Graafian follicle to corpus luteum. All right. Let's continue, guys. Describe the function of the. We are done with that. So there are 2. Name the hormone that caused the endometrium to start thickening from day 7. They're saying start. The word start. You see, endometrium is starting here. Yes. So in this case, what is the answer? In this case, the answer is. Tell me, someone saying, yeah. The answer is estrogen. Estrogen. Yeah, remember we said that estrogen, when you're discussing, said that estrogen, yeah, prepares the uterus for implantation, but progesterone maintains the pregnancy. We said estrogen prepares the uterus for implantation. How? As we ask you that question, how? By making the endometrium thick, vesicular, and granular. Start the thickening, you see? Thick, vesicular, and granular. So it is estrogen. And they're saying day 7 here. So estrogen. Name the state one other function. Name the hormone that goes, okay, we have named it. And they're saying state one other function. You see, now I've forgotten. Just gonna skip it. Yeah, because I saw Thomas say, where is the other mark coming from? Name one other. Guys, don't forget that you have boobs because of estrogen. Now I'm asking, if you are men, you have boobs, you have estrogen. I don't know. Don't ask me. Anyway, yeah, estrogen is responsible for secondary sexual characteristics in female. You must say that. Don't say secondary sexual characteristics. What about in male? I don't know. I don't know because it's not our concern now. That doesn't mean that I don't know completely. If you have one, if you have that feature, you can tell us. Yeah, you have to know yourself, guys. It's not that I'm just talking bad and what. No, man. Yes. All right. Let's continue, guys. All right. They're saying that the second function here is what is the second function? The second function is puberty. We say that it controls puberty. Secondary sexual characteristics. If you talk about boobs, you can talk about okay, we don't call them boobs, we call them mammary glands. Ne? Yeah, you can talk about your voice, the way you are talking. Check the face. Yeah, basically you have the secondary, you have the primary sexual characteristics. Yes, you have, but when you're still young, it's not yet developed. It's supposed to develop. When it's developing, we call it secondary. Now, that's why you call it secondary. But because they are linked to sex, your gender, then it becomes sexual. So it becomes secondary sexual characteristics. So that's it. Name two ovarian hormones and state their origins. When you talk about ovarian hormones, you're looking at the hormones which are produced in the ovary. Yes. Which hormone? What hormones which work in the ovary? You understand? We are looking at the hormones which are produced from the ovary. That's why even here they are saying ovarian hormones. Then here we are saying, yes, you see, even they are showing you them here. Yes. So this is follicle stimulating hormone, and then this is the uh, let me show you. This is FSH. Why am I saying FSH? LH. While this one is FSH. Then this one is what? It increases after day 14, so it is progesterone. And then this one is estrogen. Sometimes when we are in America, we say estrogen. But please don't use that one. We have an order. Yeah, we use the British English. Yeah. So I don't know, we like to speak American way, but when you're writing, you write the other way. Oh, that's Mzanzi. Anyway, let's go. So it means that these two becomes, so the answer here becomes estrogen and progesterone. Estrogen and progesterone. So estrogen and progesterone. Yes. So if I have estrogen and progesterone, what does it mean? So where does this estrogen come from? It comes from estrogen comes from here, the Graafian follicle. While progesterone comes from here, the corpus luteum. You write them. So this one comes from the Graafian follicle, or this one comes from corpus luteum. Yeah. Don't I deserve a like and a subscribe? After that, yeah, don't forget to subscribe, guys. Yeah. Ish, guys, the year is going, guys. It's just a small gospel, year is going. Don't you think I'm gonna miss you guys? Ish. Let's pray that you guys will pass, Ish. Because these books are boring, Books are boring. But if you pass, you get money. It's nice, man. I always tell my students that, oh, you want to eat like me? Read your books. Go get a job, Get money. Yes, eat whatever you want, Don't ask me why am I eating like that, or I should give you a bite. No. Go, read your books, get money, eat whatever you want, But books are boring. Yeah, they're boring. But what you need to do is to put the energy. You'll get there. All right, let's continue. 2.3. They're saying, should I read all these things? Yeah. Time. Guys, you read this. They're saying this text 1 and 2 below. Okay, answer the question. Okay, diagram below shows a contrast between the health brain left and then the health brain. This is the left. They're just trying to explain what is here, guys. Yes, this is the damaged one. Saying identify one structure problem of Alzheimer's brain mentioned in the text above. If you read, they mentioned it here. They say that in Alzheimer's brain, abnormal level of natural occurring protein clump together to form this. You see, this is what they are looking for. It forms this plague. So that is the structure they're talking about. So that was supposed to be the answer. Identify the type of neuron depicted in the diagram above. If you look at the neuron, this is the cell body, and then these are the dendrite, the axon, the terminal branches of the axon. The terminal branches of the axon. Dendrite, cell body, axon, the terminal branches of the axon. Yes. You know, how does impulse move? Dendrite, cell body, axon, the terminal branches of the dendrite, the terminal branches of the axon. Axon. Do you know how to sing that? It means that the impulse moves from dendrite, cell body, axon, to the terminal branches of the axon. That's the impulse. That's how it moves. Yes. All right. Let's continue. Identify the type of neuron depicted. So it is moroneuron. Moroneuron. Neuron. Yes. Name the membrane that surrounds the axon and state its function. The membrane that surrounds the axon. Which membrane surrounds the axon? Is Myelin sheath. Myelin sheath. I hope you wrote the correct spelling. Me, then lin, sheath. Sheath. Yeah. Name two symptoms of Alzheimer's disease. Here we can talk about the memory loss. Memory loss. And then number two, we can talk about the cognitive decline. Yes, here when we talk about cognitive decline, now it means that you can't talk that, like, think critically. Yes, if you see that your thinking capacity is decreasing, you might be suffering from that. Yeah, let's go, man, to the next question. Which question is this? This is a synapse. Yeah? You have it in the distinction material. I think you have it. Yeah, you have it in the distinction material. We discuss about it. Let's see. Diagram below shows the transmission of impulse between two neurons. Information in the passage states Alzheimer's results in the synaptic loss. Give two letters of region in the diagram which represents a synapse. Give the letter of the region which represents a synapse. Now we said a synapse, the whole thing is supposed to be a synapse. But we say that this the small space between two neurons, a junction between two neurons. So it's a junction between neurons. So I shall say that C, this small space, synaptic, this space, that's what it's called a synapse. So in this case, we're gonna say that it's C. Yes. State the significance of the synapse and identify the specific area where its loss might lead to Alzheimer's symptoms mentioned above. So here there is no passage. You understand? Yes, there is no. Is it linked to this? Let me see. It's not linked. Oh yeah. Linked. Okay, let me show you. Here we're saying that. Give the letter of the region. Okay, here in this case we have said that it's C. Let's state the significance of the synapse. I've been telling you that the function of the synapse allows impulse to move in one direction. Yeah, unidirectional flow of the impulse. Unidirectional flow of the impulse, or allows impulse to move in one direction. And then where it slows. Yes, they're saying that identify specific area where slows might lead to Alzheimer's symptom mentioned in question. Wait, question 3, 2.34, 3.4. Oh, this symptom. So basically, if we look at scientifically, we can say that it can it loss it can it you can have a loss in the hippocampus. Ne? Let me just write it here, but I think I should maybe say it in a simple way. This is what you call hippocampus. Yes, hippocampus can lead to Alzheimer. You understand? And basically, what is hippocampus in layman's language, guys? It's just a region of the brain that is associated with primary memory. The region of the brain, that's if this happens, yes, it can happen in the hippocampus, but I don't know. So in this case, we can say, because it's a part of memory, so we can say cerebrum. Yes. So I don't want to talk too much of medical. Yes, so the cerebrum. That's where hippocampus is located in that regard. So hopefully those who wrote cerebrum will get a mark, and those who wrote hippocampus, you also get a mark if at all your teacher knows it. We're not islands. It doesn't mean that we know each and everything. Yeah, it might not be included in the memo. We only wrote this, but scientifically, hippocampus is also correct. All right. Name the part of the neuron that transmits impulse to the cell body. Ah, we have just read. We say that. How does the impulse flow? Ah, sing with me. Yes. Dendrite. Dendrite. Cell body. Axon. To the terminal branches of the dendrite. Dendrite. Zone. Cell. Dendrite. Cell body. Axon. To the terminal branches of the dendrite. You understand? Yes. So now we know. We say that impulse moves from dendrite, transmits impulse to the cell body, dendrite to axon, dendrite to the cell body. So it is dendrite. You understand? Then from cell body to the axon, dendrite, cell body, axon. You see? Then to the terminal branches of the axon. So it means that transmits impulse to the cell body, transmits impulse away. That is the axon. Axon. Yes, I think guys we got it right. Yeah, thank you. We go to the next question. The next question. Yes, the next question is saying that about the eye. The diagram shows the contrast between the normal eye and the eye with the cataracts. Okay, tabulate the difference between the normal eye and the cataract. In this case, we just have to look at the differences, what you see here. So basically, here the lens is clear. Just that you guys, you have a black and white. I don't know whether the paper were photocopied or what. I don't know. I didn't see there. But if maybe they brought them from the district and then they are clear, I think you can see this clear. You understand? So basically here you have the eye is clear, and then this is the eye is cloudy. Yes, the lens is cloudy. You know the cataracts. If you are using the distinction material book, you know it very well. Yes. So we can say it has a clear lens, it has a cloudy lens. Don't forget I said tabulate. If you're tabulating, you have to draw a table, and then you say there normal and normal eye, and then here eye with the cataracts, and then here you say clear lens, and then here you say lens is cloudy. So you the way how you modify it depends on the way how you have modified it. That's number one. Then number two, you can talk about this one allows light. The normal one allows light. They say structure. Tabulate this the difference between this one. Yeah, check if you look at it here. Here we are done with it. If you look at it here, it allows light. It allows light to pass through easily. But here what happens, it scatters and blocks light. Check, it scatters. Check how many images are formed on the retina. So it scatters light. So on the side of cataracts, number two, it scatters and blocks light. Here it allows light to pass through easily. Think I'm clear there. And then we give you one mark here, one mark here. Okay, one mark there, one mark there, one mark there, and one mark for the table. It's supposed to have these two caption, and then it's supposed to be closed. Yes. Name the treatment. This one is surgery. Surgery. Yes. Name and describe. When you say name, you just have to name and then you get the mark. Name and describe the process that caused the lens to of the normal eye to adjust to the object that is seen less than 6 meters away after viewing. So in this case, you talk about where to. So in this case, where to is the you are from far object, you're going to the nearby object. If the nearby object, the lens is supposed to be thick, and if the lens is thick, then automatically the suspensor ligament must slacken. And then slacken, this one must become the tension on the ciliary muscles must contract. So in this case, you are describing the nearby object, but they said name accommodation. Accommodation. Then now you describe. We give you a mark there. Ciliary muscles. Ciliary muscles. Muscles. Muscles contract. Contract. That's number one. Number two, suspensor ligament slacken. Suspensor ligament slacken. Then the tension on the lens. Number three, tension on the lens decrease. Decrease. Ne? Meaning that now the lens becomes more. The lens becomes more. The lens becomes more convex. The lens becomes more convex. Now more the light rays are bent more to the retina. Light rays are bent more or are refracted more, because bending and refraction is the same thing. Are bent more to the retina. Are bent more. The word more must be there to the retina, so that now you are able to see a clear image is formed onto the retina. So the last point is supposed to be a clear image. Clear image. Image is formed onto the retina. So guys, basically that's it. On that, I think guys you got it. Ne? Guys, don't forget to subscribe. Ne? Yeah. Subscribe, like, share. Yeah, don't. All right, guys, let's go to the next question. Am I reading everything here? I am, man. You have to read, ne? Average million per milliliter before, during. Hey, I don't want to read this, but let me just read it because it's about men. 5 men who normally wear loose fitting underwear were selected to be part of an investigation. The 5 men in the study were asked to wear especially designed underwear that are tight fitting for 14 to 16 hours every day for 7.5 months. 7. Imagine you and it's too tight. Is it fitting you good? Yeah. Okay. 7.5 months. The test, the reason why is so that you give it enough time, for the experiment. The underwear kept scrotum and the testis very close to the body. Man, squeezing for 6 months, for 7 months. Man, right. The men, what were they given now? The men provided a sperm sample. How did they collect the sperm sample? You will tell us. I don't know. Yeah, several times leading up to test period. You can't say that, you guys. You can't say that. Go through. The chart below shows the average sperm count million per milliliter. Guys, it means that you have a lot of sperms just on a small spoon. Like 100 million sperms, man. Before, during, and after the test period. Okay, let's start. Before, check the sperm. These are millions, guys. Don't forget, these are million sperms. 300 days, man. Yeah. State independent variable. What is independent? The number of sperm will depend on the type of underwear you are putting on, whether it's a tight or it's a loose. Question: which type of underwear are you putting on? Tell us in the comment section. If you are losing, you will be looking for people to help you, to give you more manhood. That's, by the way, the sperm are not connected to manhood. Can be like a horse, but no sperms produced. Sperm counts are not linked to the manhood. All right. So the answer here, the type of underwear you are putting on, the type of underwear dressed, either loose or tight. So, and then here, because they talked about it here, yes. Then they say dependent automatically the sperm count. Sperm count. How many sperms are, yes. Beside the repeating the experiment, yes. Suggest how this investigation could be made more reliable. So basically, you have to increase the number of individuals. They not more than, not only 5. Increase the number of individuals who are subjects to investigation. Randomize your samples. You understand? Yes. You can also randomize the participant. Don't just have to make them, yeah, make let it be random. Just exchange them just like that. Yes. And beside the repeating experiment suggests, okay, we have done that. Increase the number of individuals from 5 to more individuals. Yes. And then also can talk about the randomization of the individuals. Yes. Explain how fitting underwear has an effect on sperm count. Hey guys, if you're putting on a tight underwear, yeah, man. Okay, we can say that basically when you see tight, it means that we say that, look, they say that it rises, it pushes. Where did they say? Underwear keep the scrotum and the testicles very close to the body, and the body is hot. Yes. So what does it mean? Yes. So it means that you can say by how is it affecting? So we can say that by elevating the scrotal temperature, impacting on the blood flow to the testis, hence low sperm count, or less, hence low sperm production. You understand? So putting there is another way you suppose you can just push the testes they are pushed above the body, yes, which has higher temperature above, or higher temperature above 35 degrees. Do you understand? And then this lead to low production of sperm, or denaturation of the sperms, because high temperature denatures the what? The sperms. So it means that you're going to have low sperm count. Or it might not be denaturation of the sperm, denaturation of the enzymes which are involved in the production of the sperms. Yes. So basically, high temperature lowers the sperm production. Yes, it lowers the sperm production. Then you're going to have a low sperm count. So what is the solution for that if you have low sperm count? It remains that you have to eat food in the morning. In the evening, it's not enough. You have to eat in the morning, breakfast. I don't know whether morning is breakfast, morning before you leave. Yeah, breakfast, lunchtime, after that, 4 o'clock at night, and then you wake up at night. You have to eat and eat and eat, so that by chance you can make the other person full. In other words, when he's full, becomes capital D. You know that when he's full becomes capital D? Yeah. What looks like that? Ne? Yeah, it becomes what? Capital D. Becomes what? Capital D. Anyway, that's it. Just joking. Let's go. Explain how the type. So we have said it, talked about it. Suggest why sperm count was measured before the test period. The reason why they measure the sperm count is to bring about the baseline. For comparison, yes, comparison, yes, to compare the results. We need, oh, how do you know that this effect is as a result of the tide? We have to know before and after. You understand? Yes. They're saying that determine what scientists analyzed during day 30 to this. What did they analyze? Basically, what they analyzed between 30 with 30, day 30, and then 255 around here. What they analyzed, they saw the trend. Yes, how many marks are here? You see that there is a decrease in the days since the first semen collection. There is a decrease in the number of sperms. Yes, decrease in the number. Check as the day uh increases from 30 to 90. You understand? And then now it becomes constant onwards. You understand? Or until 255, there is a decrease. So you can't quote this. You can quote this, but you can't quote this. You can't say that decrease from 60. No, no. Now you're just telling us what is on there. So there is a decrease. There is a decrease from 30 to 90. You understand? And then it becomes almost constant until 255. Or what they say, determine what the scientist analyzed. Is that's what they analyze. So basically, the scientists they analyze the trend and the change in the sperm count over time. Yes, and then they observe the impact of wearing the tight fitting underwear on the sperm count. And what is the impact is there is a decrease in the sperm count from 30, did I say from 30, from 30 to 90, and then there is a slight, there is the sperm count becomes constant to 255. So basically, that's it. Depends on your English. You have put it out. Ne? Yes. Well, question 3. Guys, I only have 15 minutes to finish this question 3. So I need to be a little bit faster. It feels like that question 2 sometimes it's a little bit complex or complicated, but that's it. Let's go. Question 3. Process A, transcription. Process B, translation. Provide the heading of this. This is the protein synthesis. Someone saying yes. Yeah, yes. Name the process. We said this one is transcription. This is translation. Eh? Yeah, so we're done with that. Then they're saying that describe the process, the role of C. Where is C? The role of C of this. They even brought in the final exam for May, June. Yes, the role of C. The role of C is it carries specific amino acid to the ribosome. You see, it is bring it there. Yeah, so it carries the specific amino acid to the ribosome, or it carries the required amino acid to the ribosome. That's it. Then they're saying that 3.1.4, okay, they're gonna be okay, RNA anticodons. They're saying these are anticodons, so we know that these are RNA. It's not DNA, it's not messenger RNA. So okay, provide a label of chemical bond D. Chemical bond D. Chemical bond D. This is this bond. This is peptide. Peptide bond. Guys, I told you this is not polypeptide. It's peptide bond. Peptide bond. I know some people wrote peptide bond. You'll get it wrong. It's supposed to be peptide bond. Provide, okay. Identify the amino acid E and F. E, when you come back here, we see E. E, since E, it has the transferrin A already, so I'm just gonna quote from there. So it is GGC. GGC, this one I quote that because they said this table here, they said it's transferrin A. So GGC, it is the proline. So F is E is proline, and then F is UGC. UGC, I come back here, I look for UGC. UGC is the threonine. So it is threonine. So that is the answer for that. Provide a DNA triplet that would have coded for G. G, where is G? We come back here, we look for G. G is here. This is messenger, so I have just to convert it. So it's gonna be G goes with the C, C goes with the G, and then G. So the answer here is gonna be CGG. Ne? Yes. This answer there is C. Did I say CGG? Yes, CGG. They are saying that during the replication, one of the nucleotide might be replaced in the wrong position and forming. I told you this one they're always bringing and you're always failing it. Okay. Forming DNA strand. Okay. Name the type of mutation. Since this is the base, so it becomes gene mutation. This paper is full of genes. Sorry, it's full of mutations. Hi. Okay, let's continue. It's full mutation. Identify DNA triplet code that resulted in code G were undergone where to undergo the type of mutation mentioned above and a triplet of this. Okay, G. If I look at this, triplet of this is DNA. So this is DNA CGT. This is DNA, ne? DNA. So I need to put it into transferrin A, and then I find out triplet G. G is this one is DNA is CGG on DNA. On DNA it is CGG. So what happened? They say that they changed it to this. So this is also DNA. So I'm gonna convert it to messenger. So it's gonna be GCC. Then I convert it to transfer. So it's gonna be GG CGG. So I look for CGG CGG. CGG is alanine. See, alanine. Yes, and then CGT I convert it into, it's gonna be GCCA, and then I convert it again. It's gonna be CGU. So I look for CGU. Yeah, is it CGU? CGU. CGU is alanine. Who's alanine? This one is alanine. So it's alanine. Even this one is alanine. Alanine. So basically, the protein will not change. That's the meaning. That's the meaning, guys. Sorry. So it means that. So how do you explain? Explain the effect that would have on the resulting protein. So this changed to this, of which both code for the same amino acid. You mentioned the amino acid. You tell us that this changed to this, yeah, of which they code for the same amino acid, alanine. Therefore, this will have no effect on the resultant protein. There will be, or the protein will remain the same because the amino acids are the same. Yeah, I told you about that. Then you go to meiosis. What is they're saying? Label A. What is A? It is homologous chromosome. B is the spindle fibers. C is the chromatid, and D is also a chromatid. Ne? Yes. So now we are again saying identify the phase mentioned above because they are in the middle. So it is metaphase, phase, but because they're in pairs, so it is one. So it is metaphase 1. So we are done with that. Saying that name and describe the process. Did I provide one visible reason for your answer? Visible reason: chromosomes, homologous chromosomes are aligned along the equator in pairs. Homologous chromosomes are aligned along the equator in pairs. That's what you're supposed to explain. Yeah. Things are going, Don't forget to subscribe, guys, like and share even to others so that they benefit, Yes. Name and describe the process that resulted in C and D being different. The process is crossing over, and then now here you talk about homologous chromosomes come together. Yes, and then the chromatid of each homologous chromosome chromatid of one homologous chromosome and another homologous chromosome, they cross over at a point called chiasma. Yes, and then after that they separate. So they exchange genetic material. Then after that they separate, so that now each chromosome have genetically different material. So you just describe the process of crossing over. Yes. State two importance of the process mentioned above. So basically, the process of state the importance of the process. It brings about genetic variation. Guys, it's better to say genetic variation. Don't just say variation. It's better to say genetic variation. How many chromosomes would normally be present in each daughter cell once the human body cell have completed the meiosis? They're saying human. The moment you talk about human body cell, then you talk about 23 chromosomes. At the end of the process of meiosis, one daughter cell had extra chromosome. Yes, describe how this might have occurred. So basically, it occurs. It is due to the failure of the chromosome 21 to separate during anaphase. You understand? It is How did it occur? It is as a result of the failure of the chromosome number or chromosome pair or chromosome number 21 to fail to separate during anaphase. Yes, so that's how you because here you're talking about the non-disjunction. Yes, so the failure of the chromosome 21 to separate during uh, uh, during anaphase. Then we go to the next question. They're saying that the table below shows the result obtained during investigation of the pure breed black mouse crossed with a mouse which is brown. They're saying the gene for black is dominant over the gene for black is dominant over gene for brown. So F1 generation were used to were then used as parents. The fourth, so they're used as so many that this second generation. They're saying that the fourth breeding were used as a result of the table. They're saying that from the data above, calculate the simplified phenotypic ratio of black. They're saying simplified phenotypic ratio. Yes. So of black mice to brown mice in F2 generation. So is this the F2 generation? Meaning that you're gonna add all of them. You're gonna say 8 plus 5 plus 3 plus 8. You get the answer, and then even here you say 0 plus 3 plus 3 plus 2. You also get the answer. You understand? Yes. Once you get the answer, now you come here. Which answer is that? I don't know. If I combine here, this is 16, and then it's also 8. So it's 20, 24. And then here is 3, 6, 8. So it's 8. So basically, I'm going to say 24 to 8, but they said the simplified phenotypic ratio should divide by 8 because 8 goes into 24. So I'm gonna get here what am I going to get here? I'm gonna get 3. It goes into 3 times by 1. So this is the answer they are looking for. Yes. Then they are saying that they provide the evidence, provide the evidence in the data collected to support that the statement, the gene for black is dominant over gene. Yes, if you look at here, the first cross, yes, first generation, all of them were black. None was. And when you see, they say that they crossed black and brown. You understand? They crossed black and brown. So when you cross black and brown, you're supposed to get both. But now they said only they said pure breed, meaning that black was capital B, brown was small b, small b. So when you cross this with this, you're gonna be capital B, small b. All of them they're gonna be capital B, capital B, small b, capital B, small b, capital B, small b. So all of them are black, which means that black is dominant over brown. So in this case, you can say the first generation, all of them were black after crossing the black and the white. So guys, and the brown, yes. So the way how you have phrased it or you have spotted another answer there, then is also fine. They're saying do a bar graph of the phenotypic ratio of F2 generation for each breeding pair. So guys, here they're just talking about the bar graph. I'm not going to draw a bar graph for you, but I'm just going to talk about what is really needed here or what you needed here to do the bar graph. So in this case, in this case you have to, this one I told you is all this is x, this I, this one is d, which means x and y. So this one is going to go on y, this is going to go on x. We use that formula of id, guys. Oh, sorry guys, use that formula of id, and then now it means that your graph is going to look like, yes, so I'm going to bring this F2 generation here, and then I'm gonna put these things here. I'm gonna put this in different sections. So I'm gonna use first, second, third, fourth. Yes, so the first, second pair, third pair, then fourth pair. Yes, but you have to write them in full, or you use a key. Then now here you have to write the highest is 8. Maybe you say 0, 2, 4, 6, 8. You see, now we start plot this, this, and this. So this one, the first one is 8, the second one is 0, so nothing you wanna put. Then this one is gonna be 5 and 3, the second one is 5, it's around there, and then the other one is what? Is 3. So this one you combine it, then you leave another space which is equal. Then this one is gonna be 3, 3, gonna be 3, and then the other one is gonna be 3 also. So you guys, you draw like that, even then you draw another, but this space and this space must be the same. Sometimes we leave this space, sometimes they don't leave. In most cases we don't penalize you that. And then now this and this must be the same size. You put the caption there, you put the unit if it's needed. Here you're supposed to write number of mice, in bracket black and brown. Says number of mice, so black and brown. Yeah, so basically that's what you're supposed to look. And then here we can gonna say title, and gonna say type of graph, title, scale, labeling axis, all those things. Then we keep giving you marks. So but basically that's the bar graph they are looking for. Let's go to the last question. And then the Drosophila melanogaster feeds on sugar found Drosophila melanogaster, we call them the fruit flies, né? Yes, fruit flies, né? That it comes on here. Yes, that one. So Drosophila melanogaster so we are saying that Drosophila melanogaster feed on sugar found in damaged fruits. Ne? They're saying that the fruit you are okay of female called wild type, and then the gray striped. Okay, the wings are longer than the abdomen. Okay, their mutant version is D. Okay, Drosophila dark. This is dark, and then this is the small vestigial. Okay, sharp. So here you have. So if you look at it, we have two things. We have the color and we have the length. Yes. So we have the black, which we call the ebony, and then we have the gray. You understand? Yes. So now they're saying define allele. Allele is an alternative form of a gene. So that one, you know, some mutant variation of Drosophila melanogaster were as a result of the different number of chromosomes in the genotype. Name the type of the mutation. They're saying, I told you, this paper is full of mutations. So because it is a full number of chromosomes, so it becomes chromosomal mutation. Chromosomal mutation. So that's the answer. Someone saying, whoa, why are you crying? Chromosomal mutation. So they're saying that when two fruit flies with wild type body color, meaning that gray, were crossed, some of the F1 generation were ebony. There are two types. This one, the wild type color, yes, which we said is gray because they said it here, it's gray. You understand? They are saying that some of them were ebony. What does it mean? It means that it was heterozygous. Use the monohybrid cross, genetic cross, to show the percentage chance of having the ebony. Yes, so in this case, guys, you have to do the P2. Yeah, because they said some of the F1 generation had ebony. Use them. Okay, it's F1. So it's P1, F1. So we give you a mark for that. Then you say phenotype. So in this case, we come here, we look for the phenotype. I told you that we get it from the question. It's wild type, which is gray color of the Drosophila melanogaster with the gray color, because they said two of them. They said two of fruit flies, wild type. You understand? Yes. So meaning that now we go to genotype. Then it's gonna be gray. Remember, gray is let's give it a gray. They said wild type is capital G, so it's gonna be capital G, is capital G. But they say they produced ebony. Ebony is the recessive allele. You understand? Yes, there is a recessive allele. Therefore, it's gonna be small g, small g. Why do we say it's a recessive allele? Because even here they showed it here. Yes. Then you're gonna say meiosis occur, and then you have capital G, small g. You're gonna have capital G, small g. And then now, because these are gametes, you circle them. Don't leave them at the naked as if at the beach. Yes. Then now we are saying that you can use the Punnett square or you can just cross. So this and that is going to be capital G, capital G. That and that is going to be capital G, small g. That and that is going to be capital G, small g. That is small g, small g, small g. And then this is a fertilization. And then these are offsprings. You understand? So now we come here, we're gonna give you some ticks there, but the compulsory mark, since they say that, show the percentage chance of having ebony. So we're gonna say that the percentage chance here, the phenotype here, basically the genotype, we know it. Okay, phenotype, 3 of them are black, are gray, and then 1 is ebony. Yes, so now we're gonna have to do 1 out of 4 because the total is 4, is only ebony, which is 1, 1, 2, 3, 4. So times 100, which gives us 25% chance of 5% chance of ebony. You understand? Yes. So basically, that's how you're supposed to do that question. Then the last question, they're saying that name the type of cross which would compare the body color with the size at the same time. This is a dihybrid cross. And then lastly, they're saying that identify the phenotype. If G, capital G, is the wild type, that is basically gray, wild.